Hi, this is a video presentation of the SHREC 2020 paper Classification in Cryo-electron tomograms, presented by me, Ilya Gubins, on behalf of the track organizers and groups from 7 universities that participated in the benchmark. First, I must explain the domain of the problem. Life exists at different scales, and we need appropriate imaging methods for different scales and goals. At its smallest, there is multiple available methods, each of which has different pros and cons. Nuclear magnetic resonance spectroscopy can obtain 3D models of biomolecules at atomic resolution, but only for small individual molecules. X-ray crystallography requires the sample to be crystallized, which changes the structure of a biomolecule. Electron microscopy with enough power can achieve atomic resolution, but high energy damages the sample and often requires chemical fixation, which again changes the structure of a biomolecule. However, an approach called cryo-electron microscopy was developed. It is a combination of different methods for which 3 scientists received the Nobel Prize in Chemistry in 2017. Jacques Debouchet proposed sample preparation method that involves cooling the sample so that it becomes vitrified, meaning that ice is formed, but the cooling was so fast that the water molecules did not have time to form ice crystals. Joachim Frank proposed image processing method to obtain 3D structure from 2-dimensional images. If you have thousands of copies of the same particle at random orientations, computer can calculate how the orientations relate to each other, match them, and generate a high-resolution structure in 3D. And finally, Richard Henderson was the first person to actually achieve atomic resolution with cryo-electron microscopy in 1990. Since then, hardware of microscopes was significantly improved, and since 2013, with introduction of a new type of detectors, researchers routinely achieve atomic resolutions. Not only the resolution is important, but the freezing itself can be done mid-process, allowing us to pause the life in the sample and visualize processes in it in situ or on-site. This is crucial to understanding life's chemistry and therefore development of new pharmaceuticals. In cryo-electron microscopy, image is formed similarly to how it is formed in any transmission electron microscopy. In short, the beam goes through the sample onto the detector, and we obtain an image, a 2D projection of the original sample. As mentioned before, there is a way to obtain 3D structure from projections of the particle, but you need to have thousands of them to achieve high resolution. There is another way to obtain 3D structure of the sample with tomographic approach. Conceptually, this is very similar to how tomograms are done in medicine, for example, MRI in hospitals. Instead of obtaining just one projection at a fixed angle, we rotate the sample around one axis, usually at -60 to +60 degrees, and obtain so-called tilt series, projections at different angles. Using back projection, we can obtain reconstruction volume of initial sample. Microscope's electrons strongly interact with the biological sample, limiting signal-to-noise ratio, and as a result, we get very noisy reconstructions. Moreover, at rotation angles of more than 60 degrees, the sample often becomes too thick to yield meaningful information, so the reconstruction will always be an ill-posed problem with missing data. Even with such noise and missing data, we can still detect particles in it. Following subtomogram averaging approach, we can extract the volumes of the particles, align and refine them, and finally average them together to obtain a high-resolution 3D structure. This approach introduces the challenge of correctly localizing and identifying particles in the noisy reconstruction. Manual segmentation is rarely feasible due to the amount of volumetric data, and automated approaches must be used. The baseline method is template matching, still widely used in groups all around the world. However, recently, machine learning has seen a successful application to Cryo-ET. The common problem is that all methods are validated and used on different tasks and different datasets, making it difficult to compare and draw conclusions about their relative performance. With this benchmark, we want to first release a novel, realistic, yet fully annotated dataset, and second, support researchers by providing tools to compare the methods. To generate the dataset, we simulate the whole tomographic pipeline. First, we generate sample. We put macromolecular complexes in the sample volume at random orientations. Next, we simulate the vitrification process. So we add ice, various artifacts, and noise. After that, they simulate electron microscopy and the effects on the sample. Finally, we add detector-dependent noise. For the benchmark, we simulated 10 tomograms with 1 nanometer per voxel resolution. We chose 12 proteins of different sizes and shapes and put 2,400 to 2,800 of them in each tomogram. We also generated annotations and provided them for 9 tomograms, leaving 1 as a test tomogram. For example, this is how the test tomogram looks like. The main goal of the benchmark is to assess method's ability to localize and classify biological particles in the tomograms. The performance is evaluated on the test tomogram, for which the ground truth is not available for participants. We used commonly adopted metrics: precision, recall, F1 score, and false negative rate, also known as miss rate. We also recorded how far the predicted center of a particle was from the ground truth center, and how many results refer to the same particles. This is the second edition of this benchmark. Since then, we improved in multiple ways. Most importantly, the dataset generation has been considerably expanded. We fixed multiple problems and added important improvements: better sample generation, better microscope simulation, and more precise ground truth data. In the future, we hope to publish a separate paper on our simulator. Finally, this year we have 7 methods proposed by 6 international research groups, instead of 6 methods by 5 groups. The first method is 3D MSD. It is a supervised approach that performs semantic segmentation. They use 64 dilated convolutional layers with rate from 1 to 16. After the reconstruction is done, the authors propose to use min-shift clustering to find particle centers. The second method is DeepFinder. It is a UNet-based semantic segmentation model. In comparison with other approaches, DeepFinder also provides tools to create your own training data. To obtain particle centers, they also used min-shift clustering. Third method, 3D ResNet, is based on residual networks. The authors used ResNet of 20 layers to do semantic segmentation, and later applied majority counting to obtain particle centers. YOPO, or You Only Pool Once, is the fourth approach. In comparison with other methods, it is trained on particle centers and does not require segmentation maps or post-processing. The N3D UNet approach is comprised of 2 models. First model is denoising the initial reconstruction, while the second is doing the actual segmentation. Denoising is done with the denoising convolutional neural network, while the segmentation is done with the UNet. Method number 6 is UMC. UMC extends UNet to produce multiple outputs. For SHREC benchmark, the trained models produced denoised reconstruction and semantic segmentation with 1 forward pass. Finally, we have also evaluated template matching as a baseline that many groups all around the world use. We used Python template matching pipeline, same that is used for processing of experimental tomograms. We generated 2 different result sets with template matching, one without any post-processing and one with basic distance and voxel count filtering. This is the cropped version of the results table of the evaluated methods. You can find the full version in the paper. First column is average distance from predicted particle center to ground truth center. Second column is recall, then precision, miss rate, and finally F1 score. Below the table is ranking by F1 score, with baseline underlined. As you can see, almost all learning-based methods perform better than the baseline. We have also evaluated classification performance by class. Below, in the ranking by average F1 score, baseline is underlined. As you can see, 3 methods achieve better performance than the baseline. The results allow us to conduct more in-depth analysis. For example, one of the most obvious conclusions that we draw from the results is that methods do not really have any problems with finding bigger particles. In this confusion matrix, with particles sorted from the smallest to the biggest, you can clearly see the correlation. It is especially noticeable for template matching. It confidently finds big particles on par with learning-based methods, but for smaller particles, the performance rapidly falls. Moreover, if we plot methods together, we can see that all of them have similar spikes and downs in the performance. Another conclusion is that learning-based methods can achieve significantly better performance than the traditional template matching, in both detecting particles and classifying them. Moreover, template matching takes very long time, even when using GPU, more than inference of all other methods. There is always more space for improvements. We can work on improving the simulator, for example by adding effects like motion blur and defocus gradient. Then we want to validate our simulator on experimental data. After it is done, we can also assess and validate template matching. Since template matching is so widely used, surely such assessment will be helpful. Finally, if you are interested, please see the paper, as there are more details than was mentioned in this presentation. This is it. Thank you very much for your attention. If you have any questions, there will be live Q&A during 3D object retrieval workshop. Otherwise, please feel free to email me.